To start by welcoming everybody to the Emerging Issues in Conservation Biology seminar sponsored by the Society for Conservation Biology. I'm Mark Schwartz. I'm editor-in-chief of Conservation Science and Practice. I'll be the host for the seminar today, given by Daniele Baisero on a paper not in press. It's early online in conservation biology. It's been up since middle of July. It's called Redefining and Mapping Global Irreplaceability with Richard Schuster and Andrew Plumtree as co-authors. And he'll be talking about that paper for the next bit. I'll ask everyone to remain muted during the talk. If you have a burning question, a clarification issue just in the middle, go ahead and drop it in the chat. I'll try and be monitoring that. And at the end of the conference, the seminar, I'd invite you to unmute yourself and ask questions. I'm sure this is a really interesting topic, and I'm sure that it is a very difficult mathematical problem to solve. So I'm anxious to hear the seminar. And so with that, I'll just hand it over. Danielle, you go ahead.
Thank you, Mark. So thank you everybody who is connected from all parts of the world. And thank you for all of those that will be listening to this talk on the recorded version. So I'm Daniele Baisero. I made this paper, Redefining and Mapping Global Irreplaceability, with Richard Schuster and Andrew Plumtree. And it's unfortunate that I don't need to spend much time on highlighting the current biodiversity crisis. The current extinction rate is believed to be 1,000 to 10,000 times the background rate that we would observe if human intervention was not in play. And this has been known for some time. Over the past decades, numerous approaches and strategies have been exposed and published to try and figure out where we should protect biodiversity, where we can reap the greatest benefit, from biodiversity hotspots to global 200 ecoregions to important bird areas and key biodiversity areas, and multiple others on top of that. But even just looking at these maps very briefly, without even going into the detail of what they represent, we can see that they all highlight very large portions of the world. And frankly, we don't really have the time and the resources to act on everything at the same time. We don't have the manpower, we don't have the money, we don't have the time, as biodiversity is getting lost day by day. It's therefore important to prioritize, which sort of leads to the first question of what is irreplaceability and why should it be redefined? Now, irreplaceability is a very simple concept. It's a binary concept. The idea is that we have something that we want to achieve, an objective, a target. And for example, we are observing a site and we ask ourselves, can we lose the site or can we not lose the site? Is the site irreplaceable or not? The question is very simple, and the answers to this question are also very simple. They're trivial. Either a site is not irreplaceable, we can achieve the target without the site, or the targets cannot be achieved without the site. In that case, the site is irreplaceable. But this binary definition is very limited, because while it's important to know which sites are truly irreplaceable, the non-irreplaceable ones can exist in a spectrum. We have sites that contain very little biological value, or sites that contain an enormous amount of biological value, but that are just short of being mathematically irreplaceable and needed in order to achieve our targets. And this spectrum between entirely not irreplaceable and entirely irreplaceable is what the metric of irreplaceability tries to measure outside of the binary definition. The concept was introduced in the mid-1990s in a paper from Pressey, Johnson, and Wilson, quite aptly called Shades of Irreplaceability. And they give two non-mathematical definitions of the idea, which are the potential contribution of a site to a reservation goal, or the extent to which the options for a representative reserve system are lost if the site is lost. I said these are non-mathematical definitions, but they also introduce an approach to measure irreplaceability. The original definition, the original approach was based on combinations of planning units. The idea is that you have a study area that you're interested in, and you subdivide it into planning units, either using raster grids, discrete global grids, or irregular planning units that maybe represent administrative boundaries. And you can measure the irreplaceability of each of these sites using the method that they originally proposed, which is based on the proportion of representative combinations in which the site is needed. And this is a mathematical definition displayed in their paper. It's quite simple, to be honest. You have all of your planning units, and they can be arranged into all possible combinations. And then you look at all of the combinations and you separate them into two piles, the representative ones and the non-representative ones, where the representative combinations are combinations of sites that achieve the targets for all species that you're currently observing into your study. Then you look at the representative combinations and you simply see in which proportion each site is needed. So you look at combination, you say, what happens if I remove site A? Are my targets still all achieved? Yes or no? And you simply measure this proportion of how many times site A, site B, site C, and so on are needed among all of the combinations that achieve your targets. The idea is, if a site is always needed, it will have a proportion of representativeness of 1, an irreplaceability of 1. If a site is never needed, it never is needed to maintain representativeness of the representative combinations. In that case, it'll have an irreplaceability of 0. This is, however, very easy to do when you have a limited number of planning units. But as the number of planning units increases, the number of combinations increases exponentially, and therefore cannot be really applied to very large exercises, particularly at national level, depending on which nation you're observing, or absolutely at global level. So what happened in the following years is that there has been a race to identify strategies to find efficient combinations, which are the ones that achieve all of our targets with the least expense. The objective of these software, such as Marxan, Zonation, and Prioritizer, are to find the most efficient solutions, and they can return measures that are irreplaceability-like, such as Marxan's selection frequency or replacement cost in Zonation and Prioritizer. But they have limitations as well. They're also limited to the exponential number of combinations problem. The results are only valid for the grid system in which they're analyzed. Adding species can change the results because these are metrics that aim to find efficient solutions. And most importantly, these are not true measures of irreplaceability, so they have some limitations in this respect. But let's look at some of these problems in detail. For example, results only valid for the grid. What does this mean? Let's imagine we have a study region and we have a study site that we're interested in, and we want to measure the irreplaceability of that site, and we call the site A. Let's say that it contains 0.5 of one species representation, one species range, for example, and that we have a target of 0.5 for that one species. One way of looking at our study region is to just look at the site and everything else. But we can also divide the study region in many other ways. There's an infinite number of grid systems that we can apply around site A that we want to study. And in each of these cases, the original definition of irreplaceability will give very different results from, just in this case, from 0.5 to 0.96. And many more complex scenarios exist with multiple species introduced, or with targets that are not so close to the threshold of the site that we're observing. Another problem is that when we're looking at a representative combination type irreplaceability, so an irreplaceability measured on a combinatorial approach, is that when we add species, we can actually change the results. So all of those irreplaceability-like metrics can give very different results for specific sites that we may be interested in. This figure on the left is an example from the prioritizer tutorial. This is Tasmania. And this is an example solution to the minimum set problem. What happens is that if we introduce a new species in our study region, for example, we study the region with a set of species, and then we study it again at a new species. What happens is that some of those species can occur outside of the solution that we originally identified. So what these efficiency-based metrics try to do is, well, they have to identify sites within the boundary of the species, because those are the ones that will be needed in order for the species to achieve its target in the solution. And in doing so, some of the biodiversity features that were targeted outside of that region will be now to be targeted inside, which will change the efficiency of all cells surrounding the new site. So some planning units may become from inefficient to efficient, and some will change from inefficient to efficient. So looking at these species, looking at these traits, it's clear that combinatorial approaches to measuring how irreplaceable a site is are not robust. They can change if we change the way we look at our study region by changing the grid, or they can change just because we suddenly start studying new species or add new species to our analysis. So what we did, we went to the drawing board and we decided what behaviors do we want a metric of irreplaceability to possess? What axioms do we want a robust measure of irreplaceability to possess? And we identified these 5 basic axioms. The first is that we want the metric to possess a baseline coherence, which means that if we are able to measure the irreplaceability of a site for a single species, we don't want that irreplaceability to be lower if we then study the same site having multiple species. So each species will set the baseline irreplaceability for the site. The second axiom is monotonic responsiveness, which basically says that if we add species to our study region, we don't want sites to have a reduction in irreplaceability. This is very collinear with the first axiom. This is because we simply identify these independently. We want the metric to have a proportional responsiveness, meaning that if to a study region, a study set analysis, we add a species with a low local irreplaceability, we don't want the irreplaceability of the site to increase more than if we added a rarer irreplaceability. We would like the site to have an intrinsic stability, meaning that if we change the boundaries of the sites out in our study region, all of the sites that are unmodified should not change in irreplaceability value. And finally, we wanted a measure that would give us bounded outputs. Bounded outputs meaning that the metric should have a predefined minimum and maximum value, because if any metric that has open bounds for the values outputs cannot be flexibly used in target setting. A threshold value of 0.9 will have very different significance if it's on a scale from 0 to 1 or if it's on a scale from 0 to 10. So having looked at these axioms, having identified these behaviors, we started thinking of how can we measure irreplaceability in such a way that these are all adhered to. And we quickly realized something, that any site, independently of the grid system that it's placed in, will have 2 threshold representation values for each species. So if a species is not present in a site, that site gives 0 contribution to the target for the species. So it's clear to say that it has an irreplaceability of 0 for the species. But on the other extreme, there's also a value beyond which the site will be irreplaceable for the species. And that threshold where the site becomes irreplaceable for the species is is this. It's R prime minus T, where R prime is the total available representation to the species, for example, the total species range, and T is the target. If we have a species that has 100 square kilometers of range, and our target is 30 square kilometers, then any site with more than 70 square kilometers will be irreplaceable. And between these two, we interpolate it linearly because outside of a grid system and independently of a grid system, this linear interpolation represents the cumulative curve under a uniform probability distribution. And we call this approach proximity to irreplaceability because we're simply measuring how close for a single species a site is to being irreplaceable. And this line has a very simple mathematical formula we can see here on the right. And we developed the metrics, the new metrics that we presented in the paper around this formula. And here are the metrics in the paper. I'm not going to go in detail, but what we can see on the left is that we have two new measures. One is feature irreplaceability, which we called alpha, and is related to the formula that we have just seen, with a couple of added caveats. For example, behavior on what the metric should return if the target is 0, or what the metric should return if the target is unachievable. On the other side, on the right-hand side, we have a site irreplaceability measure that is an aggregate probability-wise measure of alpha values. So if alpha represents the likelihood that the site will be intersected by a random representative solution for the species, beta represents the likelihood that the site will be intersected when looking at all species. So if at least one species has an irreplaceability of 1 in the site, then beta shoots to 1. Otherwise, it's a probabilistic aggregate of alpha values. And we also have an R package that has been published last week. I believe it's already downloadable from CRAN. So what we did with these new metrics is we looked at all terrestrial vertebrates available on IUCN. We looked at all amphibians, birds, mammals, and reptiles that live on land. These are about 30,000 species. And we divided the world into more than 1 million hexagonal cells following the Superfund 500 discrete global grid at level 5. These are grid cells of about 486 square kilometers that are, at least by the Key Biodiversity Area program, deemed to be manageable as individual sites. Of these, 322,000 intersect land. And we've decided to set a minimal conservation target for all of these species of 1,000 square kilometers. The 1,000 square kilometers minimal target is truly minimal for most species, particularly for large-bodied species or species with very large ranges. But the way that the beta irreplaceability measure works means that if a site is identified as irreplaceable with a certain target, if we apply a more stringent target, so if we want to protect more of the species, then beta would never be lower, which means that using this 1,000 square kilometer minimal target, we can identify a set of sites that will also be irreplaceable under more stringent targets, a set of sites that we can use to start observing the world and that we can build on top of. And what we came up with is this. This is the first global systematic map of irreplaceability for all terrestrial vertebrates. And it's obvious at first glance that the pattern of irreplaceability using our metric clearly follows the pattern of endemism and species richness, which is good. The global median irreplaceability is pretty low. Most of the world has a very low irreplaceability when looking at terrestrial vertebrates. And if we decide to consider highly irreplaceable sites, all of those that have an irreplaceability equal to or greater than 0.9, and I will come back to this very specific threshold value, we find that about 3.5% of the world contains highly irreplaceable biodiversity. About 23%, about 1/4 of these highly irreplaceable sites are outside of hotspots, which for many years we have believed to contain most biodiversity, most endangered, most endemic biodiversity with small ranges. And more troublesome are the metrics that about half of these highly irreplaceable sites are not intersected by protected areas. And about 1/3 of these are not intersected by key biodiversity areas, which are the measures that were used to evaluate how well we are protecting our planet. The Aichi Target 11 aimed at protecting 17% of the planet, where protected areas were the key metric. And the current talks, the current UN talks, are increasingly putting KBAs, key biodiversity areas, as one of the potential metrics to measure our progress in protecting the planet. And of course, the last good metric is that 1/4 is entirely uncovered by either of these. Most of the world, as I said, has a very low irreplaceability value. And we can see it on this graph. Now, what we have on the dotted line is the threshold observed on the x-axis of 0.9, which is a value that has been used a lot in literature lately, but that has never been really justified substantially. So it's used because it has been used for some time. And on the y-axis, we have the proportion of the surface of the world that would be selected. So at a threshold of 0.9, we would identify 3.5% of the world as highly irreplaceable. But if we look at different thresholds, if we look at the threshold of 0.5 or even lower, 0.25, we find that the percentage of the world that is selected that would be flagged as highly irreplaceable under those thresholds is not very high. I mean, if we select a threshold of 0.25, we select 8.8% of the world. And to put this in perspective, about 8.9% of the world is now covered by protected areas, by key biodiversity areas, and 15.2% by protected areas, with a talk of protecting 30% of the world by 2030, meaning that it's potentially possible to protect almost all of highly important, highly irreplaceable sites for biodiversity. The problem is that this biodiversity is not uniformly distributed. The different countries have highly uneven responsibilities. While the median of national medians of irreplaceability for most countries is 0.08, we do have a small number of countries that have very high responsibilities. In the figure of the right, we have highlighted 12 countries which contain individually at least 300 highly irreplaceable sites, which cumulatively add to 47.6 of all irreplaceable cells that we have flagged so far. And there are 48 countries for which at least half of the sites have an irreplaceability of 0.9, and 29 countries that are entirely covered by these cells that we identified. Now, these countries that are entirely irreplaceable, we've identified 29 of these. As we can see, they're all small countries. They're all tropical countries. I believe all of them are islands, which means that the responsibility of individual countries is vastly different. And many of the most irreplaceable countries will likely need to have international support in order to achieve all of the targets that we aim for in the future. And there is potentially the need to try and divert funds where they are more needed, rather than going with the strategy that we have seen in the past years of protecting a blanket 17% of each country individually. There are some considerations that need to be taken into account when looking at these figures. The first is that irreplaceability, independently of the approach used to measure it, is highly scale dependent. The whole world, if taken at a single unit, is irreplaceable. And the smaller we go, any one single square meter of the world is likely not irreplaceable. The second is that identifying irreplaceable sites is highly dependent on how we identify the targets. So it's important to guide the future researchers into finding significant targets for species. Other considerations and limitations of the current figures is that they're only based on 30,000 terrestrial species. These seem like a lot because no study of this kind, at least to measure replaceability, has been done before. But it only represents 20% of the species currently listed on the IUCN Red List, and estimated 3.4% of the approximately 9 million species that are estimated to exist. There are taxonomic and geographical gaps. Invertebrates, plants, seas, and oceans are currently missing. And as we said, target definitions need to be improved and need to be based on science. But we're currently in the work of expanding this analysis to about 80,000 species, which is all species that are currently mapped on the IUCN Red List. But this said, this pretty much brings to the end of the presentation. And as we can see, there is a lot of work to be done yet. Thank you.
Great. Wow. Thanks, Daniel. That's good. That's a massive undertaking. So as I mentioned earlier, I invite you to unmute yourself and ask questions or put them in the chat, and I can ask them for you if you'd like. I can kick off a question that may be on people's minds. I'm wondering about the taxonomic groups. I mean, it's really great to hear that you're expanding this to 80,000 species. And, you might be wondering, or I would be wondering, how much might this map change when doing that? And one inference you might, or one way to predict that, I guess, might be to look at the stability of this map based on the 30,000 species that you already have. So I was wondering, did you try things like doing this just for amphibians or doing it for everything but amphibians and seeing how much that the different taxonomic groups change this? Among the reasons why I find this interesting is we have lots of people who are taxonomic specialists who do take something like birds and then tell us something about the importance of different places relative to birds. And one always wonders, well, yeah, but what about things other than birds? And with this data set where you have a cross taxonomic groups, you might have an opportunity to get at some of that. So I'm wondering if you thought about that.
Absolutely. I've always believed that any global type analysis should be as taxonomically inclusive as possible. I've put it in the caveats because I'm deeply aware of this issue. To answer the question, we have not at the current stage made a separate analysis by taxonomy, but we could quite easily, and this is an excellent suggestion for follow-up analyses. The other good news is that when I mentioned we're currently looking at 80,000 species, is that we pretty much already have the results and we're analyzing them at the moment. So it's not an input, a response that I can give right now, but I will be able to introduce answers in the following months if people are interested.
Great, thanks. Okay, so I'm going to talk for just a moment while people are assembling your thoughts and asking questions, and just give everyone fair warning that I have a propensity to just be able to keep asking questions, and so I will do so if other people don't step in and ask questions. But I certainly encourage people to raise your hand or put a chat in the chat box and get your question out there. I'm sure that there's lots of people wondering something about this excellent presentation. Okay, I'm going to go with my question number 2. So I really like these 5 axioms, and they made a lot of sense to me. But as I was thinking about them, I was thinking that, well, that really, the first 2 in particular, having a baseline and not having things move down when you add new species, this suggests that what you're striving for is an absolute measure of irreplaceability, as opposed to something like a relative measure of irreplaceability. So like when you look at Marxan and you're looking at the weighting of a species in a reserve plan, that really is contingent upon what data are going in. And if you change the targets and you change the things you put in, you're changing the relative appeal of any one pixel in part of the system. And so there's this distinction. I guess I'm tempted to think of irreplaceability as a relative measure, irreplaceable relative to the other things that we could do. But I think that's the wrong way to be thinking about it, right?
Well, this has been sort of introduced already in the Pressy paper from 1994, because they introduced two concepts. The first concept is irreplaceability, and the other concept is efficiency. And my personal view is that while efficiency is strongly subject to the current knowledge that we have, or to the current state of the system that we're observing, irreplaceability has the potential to be an absolute measure, as we've shown with this metric. And the problem is that if we want to start using irreplaceability as a way of identifying what should be targeted, what should be protected, we cannot really afford to have a measure that changes depending on what we are currently measuring. We cannot have an irreplaceability measure that drops if we add 3 or 4 or 5 species to a system, because then what does it really represent? In that case, that irreplaceability measure, or that measure, would only be valid for that particular planning unit grid and for that particular set of species. Well, what we wanted to achieve was to find some way of measuring how important sites are, independently of how we divide the world, independently of the grid that we apply, which is very often arbitrary and modified throughout the analysis process. And we also wanted the measure to be a measure that improved with knowledge rather than simply changed with knowledge. So we wanted to find a measure that, as we add species, can only identify more important sites rather than just change which ones are important. And this is it's complementary to measures of efficiency, such as Marxan or prioritizer. It's not meant to replace them. It's meant to observe at the same world with a different mindset.
Yeah, good. When you looked at this, when you got the results from this map, what were the things that surprised you about it? I mean, I guess when I look at that map, I see the Andes scream out, and the South Pacific islands pop out, and bits of South Africa, Madagascar. Those things seem to make sense to me. You have, I think, I don't know if it's the Rift Valley, somewhere there in, looks like Western Uganda or Eastern Congo or something, lights up, and I was sort of surprised by this. And the neotropics in Brazil don't really light up, and I'm wondering if you could give us some insight on what surprised you about this map.
Well, initially, what surprised me was how well it mapped the current patterns that we know of endemism. And when looking at it a bit more in detail, when thinking about it, I realized that what's really being flagged up are mountain ranges and islands, which is where endemic species will tend to be congregated in greater numbers. This is not to say that there are not endemic species elsewhere, because we can see tiny spots, single cells that are highlighted because we have single endemic species throughout the world, in Central Africa, even in Central America. But in those wide uniform regions, species tend to have wider ranges, which is going to reduce the irreplaceability of any one site that you look at. While in the mountaintops, you tend to have more concentrated species and greater endemism. So it's clear that irreplaceability is a function of endemism. Yeah. Which in hindsight, makes perfect sense.
Yeah. I mean, I guess I'm thinking that you might see this map shift substantively when you add plants because of the narrow endemicity of just a lot of plant species. And I mean, that would probably be true with insects too, but we know a lot less about those.
Absolutely. There are numerous species, however, particularly insects and fungi, that are only known for a few site locations. So, for example, I can immediately say, just because I've looked at them in the past days, that the UK, United Kingdom, has a number of irreplaceable sites because of species known just for one or two sites. And I suspect that this is going to be true for most of the world, where these are going to be picked up. The other thing that I suspect is that, and further analysis will prove or disprove this, is I suspect that potentially vertebrates, plants and invertebrates, respond to endemism in different ways. I suspect that they may be endemic under different conditions, but it's something that I still have to explore. And just a thought.
Yeah. Do you worry about information bias? You know, that there's lots of people interested in birds, and probably we have, I'm going to guess, a pretty good understanding of endemicity and distributions of birds globally. But once you get to plants, we probably know quite a lot about the distributions of plants in some portions of the world, and quite a bit less in other portions of the world. And as a consequence, we may have better mapping capacity for narrowly endemic species in little studied groups in more studied regions. And so we always worry about information bias in these kinds of exercises, but this might start to crop up. I'm wondering if you're thinking about that.
Well, this is definitely a possibility. And I think that there isn't an easy solution to this problem, because knowledge will always be improving. That's the nature of knowledge. And that is why we try to stress as much as possible that what we try to identify was a measure that responded well to improved knowledge, rather than a measure that simply changed to improve knowledge. Now, some types of changes in knowledge cannot be turned around. For example, if we thought that a species was endemic, and then we finally discovered that's present in a very wide territory, then that would reduce the irreplaceability of the originally thought range of the species. But there's nothing that we can do on that. On the other hand, if we identify new species, then this measure will respond monotonically, meaning that we don't need to rerun the same analysis, but we can recalculate it only on that individual site.
Yeah. Good.
One thing that we have noticed from the preliminary analysis on all other species, that there is a lot of coastal areas are flagged up. So there are a number of irreplaceable coastlines that don't show up on this map.
Right. Yeah, that makes sense. Yeah. Other folks, we're having a sleepy Wednesday in terms of asking questions. Other folks here, it's the math that scares them away. I'll tell.
It's highly technical. It's a highly technical paper. I realize that.
Yeah. Hey, on the math, you used these two components of irreplaceability, alpha and beta, and so I'm a plant ecologist by training, and I got, it immediately stuck on me that we talk about alpha, beta, and gamma diversity, and the alpha and beta here, I don't think are analogous to the alpha and beta diversity that you would use in those kinds of diversity indices, right? So I don't know if Cressy or who just decided alpha and beta, but I wonder if you worry about confusion in that regard.
These alpha and beta came up on our paper. We defined alpha replaceability and beta replaceability to distinguish between a site's contribution to a species and a site's contribution to biodiversity on a whole when looking at all species. Because we needed a way of, we figured out that we can measure irreplaceability for single species independently, and then we can aggregate them significantly. And the alpha and beta are simply to placeholder names that rank nicely, but they're absolutely different from alpha and beta diversity and not related to them.
Yeah, good. I was once told that I should wait 7 seconds before talking to sort of encourage people who are reticent to talk to talk, and I'm very bad at that. Anybody want to open a question up? Okay, Andrew says he has to leave now because he's got kids to look after. He thought it was unfair asking questions as a co-author, which is probably true, but Andrew, nice to hear from you there. Let's talk soon. Yeah. Okay. Well, I have to say that this is really exciting work, and I before this seminar started, Daniel and I were talking about this work, and conservation biology has a high bar on novelty, and Daniel was saying that they had trouble convincing the editors of conservation biology that a method published by Pressy et al. In the mid-90s, which had never been actually applied, that there's still a lot of novelty in actually applying it. And I think that you're being invited to the seminar series and you're presenting here, I think speaks to you're being right on that argument. I think that is a huge contribution to the development of conservation science. And so I've been really happy to listen to this seminar and learn about your work, and wish you all the best in expanding this out and continuing on and thinking about what happens when you add more species to the pool, how you might, yeah. And so it's very exciting line of work.
Well, thank you.
So